A warm welcome to all of you joining us today for this exciting webinar on wildlife health in Asia. My name is Daniel and I am representing Life Trust India, which is based in Coimbatore. It's truly an honor to have such a passionate and dedicated group of wildlife veterinarians, researchers, and conservationists with us. We have more than 200 registered participants for this webinar from around the world, and we are using the different platforms. Wildlife health is a crucial pillar for conservation, especially in a region as biodiverse and dynamic as Asia. The health of wildlife is deeply intertwined with the health of ecosystems and ultimately our planet. Today we have an incredible opportunity to learn, share, and collaborate on some of the most pressing issues and innovative solutions in this field. This webinar aims to shed light on current challenges, advancements, and opportunities in wildlife health across Asia. We will be hearing from three leading experts in the field who will share their insights, experiences, and research. Whether you are working on the ground, in the lab, or in policy, we hope this discussion will inspire and equip you with new ideas and tools to advance your work. Please feel free to just share your thoughts and questions in the chat box throughout the session. We will be happy to just address as many as we can during the question and answer sessions. Your participation is what makes these discussions fun. I would like to let you know that we will be recording today's session. The recording will be made available for listening after the webinar. And the link will be shared with all the registered participants. So if you miss any part of this discussion or would like to just revise it, you will have opportunity to do it. Once again, I just thank you for joining us. Now, on behalf of Asian Elephant Support, I request Dr. Heidi Riedel to give an introduction about the Asian Elephant Support and also to just introduce the speaker. Thank you. Over to Heidi now.
Thank you, Daniel. Thank you. I appreciate it. And hello to everyone, whether it's good morning or good evening, depending on what part of the world you're joining us from. I would like to welcome everybody on behalf of Asian Elephant Support, who is co-sponsoring these webinars along with Life Trust India. This is a topic, veterinary expertise has been a topic that Asian Elephant Support has been interested in for quite some time. And we have organized a number of workshops and also webinars, not just on elephants, but on other Asian species. So today you're going to hear some presentations that are a little bit broader in nature. And so without wasting any more time, I'll introduce our first speaker. Our first speaker is Dr. Meenakshi Nagendran, who is a wildlife conservation biologist, an academic, and a veterinarian. Dr. Nagendran is currently an adjunct faculty at Cuesta College in California. She retired from the U.S. Fish and Wildlife Service in 2023. At the Fish and Wildlife Service, she worked as a wildlife biologist from March 2005 in the Division of International Conservation, as a program officer for the Asian Elephant Conservation Fund, followed by the Rhinoceros and Tiger Fund, and the Great Ape Conservation Fund for Asia. Prior to joining Fish and Wildlife Service, Dr. Nagendran was in academia in California and served as Director of Bird Conservation in the California Audubon. For many years, she collaborated with international partners on field research and conservation activities in Siberia, Kazakhstan, Japan, the Russian Far East, India, US, and Bulgaria. Many of the projects included satellite tracking several species of endangered cranes, storks, and waterfowl, documenting their migration routes for the first time. She received her bachelor's in science from India, her master's from South Dakota State University in wildlife science, and her PhD from North Dakota State University in zoology, both behavior and conservation biology. And she carried out postdoctoral research at the University of Tokyo. Dr. Nagendran received her DVM from UC Davis in California. Wildlife conservation and ecosystem health continue to be central to Dr. Nagendran's interests, and her presentation today is on the topic of disease investigations in a wildlife conservation context. So Dr. Nagendran, over to you.
Thank you, Heidi. Will Daniel be projecting the slides for me, or should I do that myself?
Maybe you can just do it from your side.
Okay, let me see.
Yeah.
Let me see. I don't want to mess it up. Okay. Does that look right?
Yes, we can see it.
Yes.
Okay.
Start the
Cool.
slideshow.
All right. Yeah. All right. Thank you. Hello, everyone. So today's presentation from my side is going to be more from a generalist approach. And give me a second, please. I need to make this screen smaller for me. Okay, yeah, that works. Shoot. And what I'm going to be talking about is many things related to investigating wildlife diseases or wildlife events that occur in the field where your approach would have to be more like a forensic scientist almost. So typically with domestic animals, you would follow a case, you take a methodical approach, you would do your subjective, your objective, your assessment, and your plan, and that would also probably be there to some extent, but you need to do more when it comes to wildlife disease investigations, simply because these wild animals are in the public domain. They are most likely managed by a government authority. It could be endangered species. It could be a case where there may be some illegal activity that has resulted in their death. So the records you maintain, the detailed notes you take, the chain of custody that you must keep, these are all very, very important. So you can't just be a veterinarian, but you also need to be looking at the possibility of this going to court. There is the aspect of helping animals that are in difficulty trying to survive, but then there's the aspect of looking at many dead animals that potentially could end up requiring the courts to decide what's going on. If people are present in the area, you need to ask them a set of questions. You need to ask them many questions, and that would give you some idea of what might be going on. If nobody is present, then it becomes even more challenging. And so the way to approach some of this, and perhaps many of you have already done this, and this is not anything new for you, so I apologize if this is redundant, but oftentimes when you get in the field there, you know, if nobody's there, then you need to be thinking about what is it that I need to take samples of. It becomes very important that as a veterinarian who works with wildlife, that you also establish collaborative relationships with wildlife biologists, wildlife ecologists, environmental scientists, policymakers, etc. It's really, really important. This is not just a fancy or, you know, this is not just a research project, but this is an investment for identifying potentially what could be causing a very major problem. The questions to ask. There are lots of questions to ask. What are the species that are dying? How many have died? When did they first appear? When Is there any kind of age, sex predilection? What is the signalment? What are the symptoms of the dying animals? If they're still dying, is it possible that you can tell? Is there any kind of pathognomonic sign that these animals are indicating they're dying from? Are they dying slowly? Are they dying fast? And another very important thing potentially is, are the carcasses scattered or are they all concentrated? So, and as I continue along, I'll tell you some of the, it'll be more of a story form. So this is not my work. I'm presenting from other people's work. So it's just very interesting how things have been identified. And another thing is, if you see something that looks very novel, then you want to see, has this occurred elsewhere? That is really helpful to look at the literature and see, or even just from hearsay, has someone else seen this happen somewhere nearby, far away, in another country, in similar biogeographic region? Then, did the animals, was this a toxin possibly? Were they struggling? Was it a neurotoxic situation? What might have been some of the climatic conditions before this event? Was there a freeze? Was there a hot spell? What are the habitat conditions? And what does it look like in terms of, does the vegetation look like it might be, is there any poisonous plant around there? More questions to ask. What were they eating? Did they eat something somewhere and come and die here? Water source. It might seem like, oh, the water looks clean, but is there some kind of a toxin that perhaps was upstream and it came down, and that's the result that, a whole bunch of fish are dying here, but this water looks clean. So what does that require you to do then? Did somebody spray the area? Was there any kind of land clearing? And with respect to poisoning, was it intentional or unintentional? And we have so many stories of intentional poisoning, and then so many incidents of non-target species dying. So that's across the world, you see that. What about domestic animal disease? Okay. So there are lots and lots of things, but also remember, if there are people there, get their opinion too of what they might think is going on. And that'll help you as well. And then you may also be formulating some kind of an opinion. So be sure to register those things. If your animal is small enough, you should take the entire animal, collect the entire carcass, of course. If it's really large, like an elephant, you're not going to be able to take it. So field necropsy, field sampling is really, really important. I also wanted to tell all of you right now that, again, perhaps you know all of this, and so you don't need any of my slides. But if you do, I am providing it as a PDF, and so you can have that as well. So you don't need to take notes. What are the samples that you need to collect? Well, here is just a general slide of what you might need to do for histopath, for toxic, for micro, for parasitology, hematology, etc. And I did want to indicate that when I say a quarter inch, this is quarter thickness, that's quarter inch thickness. What are some of the standards that we use? And basically, the reason I put this together was, and I all got it from elsewhere, it's not my own, is when I first started collecting samples, I wish I had had access to something like this, because it's really important that when you gather samples, that you can use them when you come back to the lab. Otherwise, it's pretty useless. And it's also really important to know how you need to preserve them and how you need to handle them, and you need to worry about not rupturing your red blood cells when you're gathering for serology. So these are things. I wasn't a veterinarian for some of the work that I did at that time, and I was just collecting samples. But as I became a vet, I realized the importance of some of these things. So if you're working with a wildlife biologist, then your skill sets as veterinarians become really important for these aspects as well. Now let's go through some different cases or different scenarios here. For toxins or toxicosis, organophosphates and carbamates are found in pesticides which are acutely toxic, and they're used quite extensively. Some of those are banned here, but they're not banned around the world. And they're poisonous to all animals. And you find that when they're used very specifically for one species, it may not be the case that that's all that is killed. You can have a situation where there are multiple non-target species that are affected by pesticides. So we have poisoning of cattle to, say, kill feral dogs, which doesn't stop there, and then something comes and eats those feral dogs, and they die. We've had this happen. I know of cases where vultures are eating poisoned dogs, and the intention was not to kill vultures, but they're eating those poisoned dogs and dying. We've had cases of vultures being intentionally poisoned because poachers did not want the vultures to show the forest guards where their poached animal was. So they would poison after they had poached the elephants. This was this happens, I think it still happens in Africa, elephants or rhinos. They lace it with poison, possibly organophosphates, possibly something else, and you have vultures dying. And so, you have a bunch of dead vultures, but they're discovered much later than you would be able to find a poacher. So these are important situations. You need to know what is going on. It's also very important to keep complete field records, because these records will eventually likely become part of a court case, and accuracy is really important. Chain of custody is really important. Sampling should be done properly and labeled properly. Sometimes you get samples that nobody knows where it was collected, what it is, and it just gets thrown out, and it's a wasted effort. DDT, organochlorine, the kinds of tissues you would need to take. We've had, basically it's a fat-soluble, lipid-soluble toxin, pesticide, and there are lots of ramifications. It results in eggshell thinning in raptors, and for decades this was going on without people knowing what was happening, and then eventually it was discovered that this was because of the biomagnification of eagles and falcons consuming other species, and so basically got magnified along the food chain and resulted in thinning of eggshells, and eventually it was banned in this country, but it's not, I don't believe it's banned in other countries, so this problem perhaps still persists in other countries. Oil spills. This one I have particular experience with, and it was pretty devastating. I was deployed on the oil spill work that happened in 2010 in the US, the largest spill that we experienced, the BP oil spill, and it was pretty devastating. So you've got oiled birds that can't fly, that are constantly trying to get rid of this thing that's coating their feathers, and it's toxic. So we were gathering a lot of the birds, pelicans, all kinds of waterfowl, shorebirds, and of course there were many dead ones too. But then you bring them to a rehabbing facility or other veterinarians who would be doing with it. They had the task of cleaning the birds with just what they typically use in this country is Dawn dish soap, and making sure that they nourish the birds, they provide them with supportive care. So we were doing a lot of that, and there was just enormous amounts of oil on the Gulf there near Louisiana. But what was very interesting to me, I spent 5 weeks every day from dawn till dusk working on this project with literally dozens of other biologists and from across the country. And the reporting, the gathering of data, because I was a government employee at the time, and this, it was really important. The government was going to bring this case against BP. It was very, very important that we make sure that when we gather the data, that whether it's a dead bird or dead eggs or whatever, that we meticulously document where we got it from, the time of day, and make sure that the labeling was correct, and make sure that it was put in its own individual packet, and make sure that the chain of custody was maintained, because this was going to be part of the case, the court case that would then eventually, if it was true that all these birds and these eggs, etc., had died from this particular oil that was leaking from the well, they would be fined. And there was a huge criminal case, and it was a huge payout. But we were part of the team that provided the evidence. And for us, it was really crucially important to maintain everything very meticulously. I recall one evening, while we were checking nesting islands and for 2 or 3 days at a time, and one evening when I reported up to what they call the incident command, and it was everybody was talking about what it is that they had done. Not everybody was involved in biology. They were doing different things. There were oil people there, and so when it was my turn and I had to report what we found dead, all the different things, and I said, I think about 600 eggs, then there was dead silence in the room, and everybody turned around to look at me like, who is this creature that came up with this number? What are they talking about? What the heck is that? And it was very surprising for me, because the days before then, nobody cared what I said, because it was like one bird here, 2 eggs there, and all of a sudden it was like 600 eggs. And they were all part of the case. It went up to our forensic lab, the National Wildlife Forensic Lab, and they had to then get the signatures for the oil to make sure that the oil that had been responsible for that many eggs to die or not hatch, wild bird eggs, were indeed from this spill. So you can see the importance of making sure that everything is gathered in a way and maintained in a way that data are not compromised. Of course, in cases where you have animals that keep going back to the area, one of the simplest ways is to just haze them, to get them not go there. It's really difficult to contain spills in large bodies of water, especially the ocean. So talking about aflatoxins, mycotoxins, aspergillus, this is a big problem. It happens often in many places in the world where you have grains, peanuts, birds that feed there, large numbers of birds eat it, and they do die. And so this is something that, you know, if you let grains just stain on the ground and it's moist and warm, and so these are cyclical. This happens, but it's important to recognize what it is. Algal blooms. This is a very interesting situation. So these are also, they go through periodic cycles, and warm weather can create the situation where the bacteria produce these toxin, the cyanobacter, and it can be deadly for all sorts of species, and mammals in particular. We've had in the US, we've had, you know, dogs might go on, people's pets may swim and eat stuff or even drink the water in some of these areas and actually get very sick and die. We've had birds actually, obviously, die as well. And it's very fascinating. We had, this was several years ago, I think it was in maybe 2012 or 2013, when more than 300 elephants in Botswana were found dead all of a sudden in an area which they were all, I think it was in one little ponded area or wetland, and the debate was going on, what could it be, what could it be, what could it be? And so a situation like this, it's really important when you discover the animal that you do a lot of environmental sampling as well. Eventually, yes, they said it was cyanobacter ingestion and toxicity that killed over 300 elephants. I believe something similar happened in Nepal as well, where they had several rhinos, not this many, but several rhinos died from cyanobacter toxicity as well, I believe. But again, you know, you might be just a specialist for, you might be a veterinarian, but without any experience in sampling the environment itself. But you don't have to be an expert to work with others. You can, you know, you can establish collaborations with, especially if you're associated with the university, you can establish collaborations with a biologist or a, you know, environmental scientist, or you need to make sure that you get all the relevant samples so you can have, you can come up with a proper analysis of what's going on. Again, in the US, there are times when they will post signs to indicate to the public that the waters are dangerous. They also tell you, for example, if you go shellfish fishing, like oysters and things like that, that certain times of year it's very dangerous. Don't harvest it, don't eat it, because you will die. And so that's a problem. This is not just restricted to the US, even though I'm telling you of cases here, this is worldwide. There's another toxicity that's actually very common here, domoic acid toxicity, which leaves animals disoriented. You can have, you know, a sea lion show up in your backyard several miles away from the ocean because it's consumed these toxic shellfish, and they don't know where they are. There's seizures. This happens with birds too. So these are all cyclical again, and potentially dangerous, and at least for people, you need to be aware of it. And if you start to see animals thrashing around, be aware of something like this might be going on. Here, parasitic pathogens. There's Toxoplasma gondii, which is the definitive host is cats, and this is also worldwide. And we've seen toxoplasma impact otters, sea otters in the U.S., actually result in their death. And so marine mammals can be an indication, a good sentinel of Toxoplasma gondii contamination. And you can also find this elsewhere in the world. Toxo is problematic for pregnant women. They could abort. So, and it's found in domestic cats. Here it's primarily also feral cats that are roaming around. So they do the toxo. Oocytes survive a long time, and they don't seem to disintegrate. So we got multiple viral diseases, and I don't think I want to get into all of that, but there are just tons of them, clearly. And they, some are more dangerous than others. And then there's our classic bird flu now, H5N1 in particular, which is fatal to most species, birds and mammals. And here we're starting to see that it's, you've got large numbers of marine mammals dying from bird flu. You've got seabirds dying from bird flu. You've got birds of prey dying from bird flu. You've got people getting sick from bird flu. And it's also reached Antarctica, so it's everywhere. And unfortunately, it's a huge problem. And so the cycle is being well maintained. And I do have to say that, the highly congested environment in which we, in this country in particular, we practice our agriculture, where cattle and chickens are all kept in large numbers in enclosed areas. This is a great way for it to be transmitted between animals and for them to get sick. And now we've got people who handle them get sick, and so unfortunately they don't want to use vaccines for this. So it's becoming it's a big problem. Anthrax, and I don't think anybody in Asia is unfamiliar with anthrax. So yeah, it's a problem. There's a way to prevent it. Unfortunately, you can't do much with wildlife. You can't vaccinate all of them, but certainly I suppose you could selectively vaccinate a few animals if they're critically endangered. But sanitation, especially if you've got animals in conditions or in captivity, then you could certainly vaccinate for anthrax. There's an interesting case here, and I wanted to share this. This was fascinating. I was at this lab, and I was asked if I could tell what these birds died from, what these geese died from. And it was really impossible for me to look at it and say, no idea. I couldn't tell. And I just thought, well, was it a toxic situation? Was it toxicology? Or was it, what was it? And interestingly enough, these geese had been struck by lightning as they were landing on this lake. And there was no way to tell other than using this alternate light source. And it was a UV light and it was fascinating. You could tell by shining it on the wing feathers that it was not obvious. So it took them a long time to figure out. So there's another thing, you find a whole bunch of dead animals, but you can't really tell what it was because the surrounding area looks fine, but perhaps they came in when there was a lightning storm and that's how they died. And so be prepared for something like that as well. And I will end this with the case of zoonotic diseases where species jump, where disease pathogens jump from species to species. And I've just been following or watching this workshop. This is just wonderful. And if you have the time, it's multiple hours of zoonotic disease, patient zero, preventing patient zero. And if you have the time, I would encourage you to watch that, and hopefully you have access to it. It's through the National Academy of Sciences. And they talk about how complex it is. It's easy to say that, we need to go in there and get rid of this wildlife and get rid of that wildlife, or this is the problem. But when you listen to it, you'll realize that it's not so easily done, not so easily defined, not so easily diagnosed. And the complexity of disease dynamics and disease ecology is fascinating for sure. So on that note, I will end my presentation and take any questions if you have them now, or
Yes, Mini, we have a few questions. I
sure.
think one question. Thank you so much for the presentation on disease investigation in wildlife conservation context. There is one question from Dr. Azad. Can we use hematological machine used for domestic animal or pets for wild or zoo animals, or is a specific hematology machine for zoo and wild animals we use? Yeah, that's a question.
I think you can. I think it's just, if you, for example, I believe if you had, if you were looking at, say, if you use a machine for chickens, you could certainly use that for other avian species. So there are, I think it's just the settings. Absolutely, you could use some of those machines. They're not very specific to a species per se. Not all of them. Well, thank you.
Yeah, thank you, Dr. Mini. Now hand over the session to Heidi to introduce the next speaker.
Okay, thank you. And thank you very much, Minnie. We appreciate your presentation. And we'll move on now to the next presenter. The next presenter is Dr. Bahad Baviskar, who is a wildlife veterinarian with 20 years of experience in conservation medicine. Dr. Bahad is currently working as president of the Society for Wildlife Conservation, Education, and Research. His research is focused on diseases of free-ranging wildlife. With the One Health approach, he's involved in standardization of practices for health surveillance. And today his presentation will be about implementation of One Health with reference to study of disease prevalence. So, Dr. Bahad, it's up to you now.
Thank you, Edu. And welcome you all. Namaskar, everyone. So, I hope my slides are visible and I'm audible.
Yes.
Yes, great. So, as already Dr. Mini has covered the topic that veterinarian perspective in One Health and disease investigation. To move on further, the same thing I would experiences that we have faced during implementation of One Health with reference to particularly study of disease prevalence in wildlife areas. So, as an organization, when we started rescue and rehabilitation work of the wild animals, we used to receive so many cases of different species from, it was more than almost 400 species, birds, reptiles, and mammals, and everything. Most of it was included as urban wildlife. But when we started doing some research with the help of veterinary colleges, institutions, and some of the disease laboratories, so we came to know that data regarding the diseases is lacking a lot. There is no detailed study which is happening. At that time, actually, there was no study in particularly was being conducted with focus on wildlife diseases. There were occasional reports and there were occasional incidences of some of the mortalities. They used to broadcast these animals to pathology department, conduct the post-mortem examination, collect the samples, and make it as a confirmatory diagnosis that okay, this animal died of particular disease. But there was no compilation. I mean, there were few veterinarians who were exclusively working on animals and zoo animals, particularly reporting diseases. But with changing scenario as an organization, there was adaptive strategy from our side that we need to start working on One Health implementation when we are trying to, contribute from our side as a veterinarian. So basically, when we started thinking about One Health, and even I strongly believe that One Health approach is in still nascent stage. What exactly to be done on ground? How the first step should be taken? Rather, what should be the first step? And there are so many things that are broadly covered in One Health approach that it becomes rather confusing to where to start with. So basically, when we talk about One Health, it's definitely a multisectoral transdisciplinary approach, multisectoral collaborative approach that deals human, animal, and environment. So no need to talk about COVID-19, which shook the world completely, and it shook all three interfaces, human, animal, and environment, where everything was halted. So COVID is one of the example, but there are many other diseases, including rabies, including many other zoonotic diseases that are affecting. Some of the diseases are not affecting the human being, but they are affecting domestic animals and wild animals are being transmitted vice versa. So when we start dealing One Health approach, that includes number of diseases happening that are being found on the ground, in the animal, in the individual, at species level, at habitat level. There are many environmental hazards that includes climate change, flood, natural calamities, and several environmental issues that includes conflict mitigation, man-animal negative interactions that are affecting One Health. So when we talk about One Health as a veterinarian, my specific focus is more on disease prevalence. That is what I have been doing, and trying to simulate some of the theoretical models on ground. So when we talk about disease prevalence, it would be certainly a number of cases, total number of cases that are being found in particular area in the given population at a specific time. And it is very important epidemiological matrix nowadays when we are in a position to talk about One Health implementation from veterinary perspective. So we need to understand what exactly that disease is causing the havoc, what is the burden of that disease on ecology, and what are the resources that are affecting the disease transmission and disease prevention and control and eradication strategies. So it's a holistic approach that we need to consider on a bigger picture. But when we work on the ground, I absolutely understand that bigger picture is something that looks very fantasized and romanticized idea. But when on your ground, you have to focus exclusively on one individual or one species at a time. And there are a number of parameters that will decide our groundwork. But if we talk about disease prevalence study and modeling it, So these are some of the terminologies that we should use. Point prevalence, that any occurrence of any disease at particular time, at particular place. Period prevalence, that would be variation among visibility of the disease and its clinical manifestation. And another prevalence would be lifetime. There would be many diseases that will be merging and upmerging, and again and again they are coming in visibility with clinical manifestation, with lot of mortality of animals, individuals, including multi-species model. So this disease prevalence has to be studied. Unfortunately, with lack of resources, lack of funding, and lack of technical and skilled manpower, many of the time we could not focus on study of disease prevalence. So there are confusions, there are biasness about implementation of disease prevalence or disease surveillance modeling, how to go with it, whether we should start in free-ranging wild animals, whether it is possible, whether it is feasible, and whether it is required. I mean, one or two mortalities in wildlife with particular disease is absolutely okay if it's going to be natural. But if the disease is spreading unknowingly, affecting multi-species, and if we do not have any idea about the disease transmission, where disease sources, where it's coming from, how much is the extent of that disease, then in that case, on ground situation, it's going to be very difficult to prevent any outbreak if it happens. So what we started as a surveillance system, that we were reporting some of the cases that were brought to us for postmortem, or the rescued cases which were being investigated thoroughly. And we started one active wildlife health surveillance model in TCP, which is Tiger Conservation Action Plan, which is mandatory for most of the tiger reserves to be followed. So unless you change some of the things in policy, the implementation gets difficult. So what we did, we started making contact with some of the field directors of different tiger reserves in India, with more focus on central India, where we started implementation of wildlife health surveillance in TCP. So what it should be, how it should be done. So there was a lot of debate on it, how it should be done. So we had suggested, and based on the available literature, active and passive surveillance are the two strategies that can be effectively implemented. So we started working on buffer villages of the tiger reserves, where we knew it that there is a lot of interface between domestic animals and the wild animals. So collecting the samples from the domestic animals would be obviously a lot easier as compared to collecting it from wild animals. And there are many legal issues as well, that we need to get the approval of Chief Wildlife Warden or the authority of the Forest Department to collect the samples from wild animals, especially free-ranging wild animals, where they are not rescued, they are not accidentally coming in your treatment facility. So in that case, we have to rely on the passive surveillance completely. When we are getting some of the cases, in the picture is one of the Hanuman langur, which was found positive with rabies, and it has history of biting 18-20 people in around the tiger reserve. So now active surveillance includes the proactive approach, where we go in the villages or even in the free-ranging wildlife, we collect the samples, and there are many sampling techniques what we have to rely on. So that we will discuss again. So when we started this designing of prevalence study, as I mentioned earlier, that active surveillance and passive surveillance was done in simultaneously phases. So that included a lot of things. I mean, when we talk about disease prevalence as a wildlife veterinarian, I am of the opinion, and of the strong opinion rather, that we need to understand the basic wildlife biology, the basic biology of the species that we are going to interact. So, for example, if we are talking about the raptors, and if there are some, as earlier Dr. Meena was saying, that there was some toxicity cases, including botulism. So we need to understand what these species are doing. So there are certain models that we use, like asking 5 to 6 questions. Who is the target species that is being affected, or who is that individual, belong to which species? What that includes, what are the symptoms, what are the problem, what are the situations on ground that that individual or that species is facing. Where that includes spatial distribution, whether that is being found in the water body or in the free ranging wildlife or in the interface or in the villages or in the urban wildlife scenario. When it's happening, whether it's happening periodically in particular season, or during the particular hours of the day. So that affects, for example, I would say there are many blood parasitic diseases which show some of the symptoms of fever in particular hours of the day. There are some diseases which are very seasonal, like avian cholera, avian botulism, echinococcus, which is found in certain areas with moist conditions. So we need to consider all these factors when we design the study. And we need to have the basic understanding of the biology of that species. So here we were collecting the samples for canine distemper and rabies and many other diseases. We used to collect it for RT-PCR for antigen testing and serum collection for antibody testing for canine distemper in some of the tiger reserves in central India. So as earlier said, again, I would emphasize again the same thing. When we start modeling of disease prevalence, when you start collecting the samples, I absolutely understand the ground situations are really challenging. When we collect the samples, there are many lacunas. Many field veterinarians or in the frontline staff of the forest department or even from the animal husbandry department do not know the standardization of the sample collection for different testing. For example, if we are going to collect the samples for rabies, how to collect the samples, how to collect the brain samples. Many times it happens that we have asked the vets on ground that you just collect entire animal and just send it to lab. If it's small, that's okay. But if it's big, then it's quite challenging. You have to collect the brain tissue samples. You have to collect something that really does matter. So when we talk about disease prevalence, we were saying that understanding basic biology or wildlife biology of that species, or you need to have some understanding of the habitat. So I would quote one of the example where there was mortality of the raptors in particular water body, and ultimately it was found that the raptors were dying of avian macular myelopathy, which is found in particularly raptors in for a very brief period of time. For your information, the disease causes some non-inflammatory spongy degenerative changes in the white matter of the central nervous system that was affecting the raptors and some of the birds which were eating some plant material like coots. So now if we look at the one individual and try to investigate the case on the basis of the mortality pattern, then it's quite challenging if you entirely focus on raptors, particularly eagles. So in this case, when the birds were dying, the mortality pattern was very, very short at very constant location. So that sample collection was very crucial, where the birds were collected, the post-mortem was performed, the spongy degenerative changes were the commonest clinical manifestation, rather pathogenic lesion in all the animals, all the birds. So when investigation, we made some contact with the biologist and some of the botanists. So the interaction, as we said earlier, that it should be collaborative approach, it should be multidisciplinary approach. So when it was discussed with some of the botanists, it was confirmed that it was the prime reason of the transmission was the hydrilla, because it was found that most of the leaf surface of this hydrilla was covered by the cyanobacterium, epiphytic cyanobacterium species, which caused certain nervous disorders in the birds and which were eaten by the raptors. So if you look at entire cycle, it's just a cascade of one event, and it is being followed by another. So that sample collection is very crucial. And when we are talking about prevention strategies dealing disease prevalence, then we need to understand the biotic and abiotic factors that are affecting that disease transmission. For example, some of the geese species are being affected by leukocytozoan species, and the vector of this disease is black flies. So if we want to control the spread of this disease in geese, we need to understand the basic biology about the black flies, which are the carrier, which are the vector for this disease. So they do have that foraging sites of less than 5 meters. So if we keep the birds in the cages which are more than 2 to 5 meters height, the prevalence of this leukocytosis was found to be very low. The cases were very low. So what I mean to say is that there are a number of factors that are affecting the disease transmission. So while collecting a sample from the wild animals particularly, it's very crucial that we need to understand the biology of that species. Having said that, there are multiple factors affecting that sample collection as well, like for example altitude, height, mountain, or the water bodies, presence of water bodies, or even the climate change. For example, earlier speaker was talking about the botulism. So botulism is happening in many water bodies where the birds are dying in very big number. But if we follow some of the factors and follow the history, then we will come to know that rainfall is one of the factors that is definitely affecting the cases of botulism. And not just that, there are stormy weather which has caused storms, may have increased the mortality of the birds. And as we know, mortality bird or recovery of the carcass is a very poor indicator of the wildlife mortalities that are taken into consideration. As statistical modeling, it's like not more than 1% carcasses are recovered in bigger mortality. So it's a big issue, very big issue. So sample collection has to be done very, very, very cautiously, and there are a number of steps that has to be followed rigorously, and they are lacking on the ground situation when we try to collect the samples, particularly for investigation, and even in some of the sensitive cases like forensic investigation, where the animals needs, like a tiger or leopard, needs some specification in the investigation. India do follow some of the guidelines formulated by National Tiger Conservation Authority, and every case of the Schedule 1 species has to be dealt like as unnatural death rather. It should be investigated properly. There are certain guidelines that says that every sample of tiger or leopard should be collected for rabies as well. But when on ground situation we start working, there are many resources which are lacking, which makes the entire investigation and sampling and disease prevalence study very difficult, and rather I would say impossible to get the results on. But again, I mean, when we were talking about some of the disease investigation laboratories, including LACONS, CCNB, forensic labs, regional forensic labs, Wildlife Institute of India, so we came to know that there is large gap between sample collection strategies, the preservatives that are being used for sample testing, the methods of transportation, maintenance of the cold chain, and documentation, proper documentation, verification of the validation of the documents that has to be done. Then when we submit the samples to the lab, that lab gives some reports, and on the basis of that we need to give the interpretation to the concerned authorities, and it has to be analyzed. So as a beginner, as an organization, when we started in 2000, we were facing a lot of problems: where to send the samples, how to send the samples, what should be the standard protocol to send the samples, whether it should be same for every disease investigation. Then gradually we came to know that okay, every situation might be requiring different protocol. So now we have standardized the protocol for dealing mortalities of Schedule 1 species like tigers and leopards, where we perform at least 5 to 6 sample collection for testing, for example, bacterial, viral, parasitological examination. Then we also conduct it for toxicology, and then we also conduct for DNA analysis, where the lab reports has to be validated that this animal was a tiger, whether it was male or female or approximate age, and specification like what landscape that belongs to. So in short, when we execute and design and execute the disease prevalence study, we need to be very, very, very peculiar, very specific about the protocol that we are going to follow. You can see one of the case of langur, which was again rabies positive. You can see the deformities in antlers and warts in this tiger. So in some cases we can collect the samples very easily if they are brought to transit facility or the treatment facility. But in some of the free-ranging wildlife, it was really challenging to get the permission to execute the sample collection, to restrain the animal and then collect the sample. So I understand that there are many challenges on ground, but we need to be very specific, and we understand the standard operating procedures for sample collection and its preservation and transportation. When we were talking about vultures, again, I mean, we have been working on vulture conservation for a very long time, and I'm fortunate that we could, save some of the areas where there was a lot of decline in vulture population, but because of support from the forest department, local authorities, and villagers particularly, we could revive the population some of the places. So again, I mean, as a one health practitioner, when we were talking about, understanding the basic biology of the species we are dealing, so vulture is one of the classical example that made us realize that we need to understand the nature and the consequences in different ways. So as we all know that during 90s, vulture population was declining a lot, and by 2000 it was almost 99% decline, the dramatic decline ever happening any single species because of non-steroidal anti-inflammatory drugs that were being used in veterinary practice. So when we talk about the vulture mortality, NSAIDs are the major culprit: diclofenac, aceclofenac, ketoprofen, nimesulide. These are the major culprits that are being found toxic to vulture in lab as well and in experimental studies. But besides that, the same era was witnessing the upcoming meat industry with globalization, and it caused lack of food availability to the vultures, not just vultures, many other scavenger species as well. There are many reasons that are causing declining vulture population, including poisoning. But diclofenac was a major culprit when it was found during the studies that it is affecting more than 99% of the population in Southeast Asia. With decline in the vulture population, there was no one at that time when to take place of vultures in wildlife. Unfortunately, feral dog population was also increasing at the same time, and that increased up to certain level that India started witnessing very high rise in rabies cases. So it could be a hypothesis, it could be just a logical thinking that with decline in the vulture population, are we witnessing increase in feral dog population, which results into increased rabies cases. And unfortunately, India is being called as rabies capital of the world, where we are finding more than 80,000 cases per year. So when we talk about vulture poisoning, vulture toxicity rather, then the cascade, if you can see, is something that is affecting one health globally. Southeast Asia is the prime example of this rabies, but I'm sure, and the data is also visible, that many other countries, including Africa, South African continent, those countries are witnessing rabies as well. So, my say is that when one species decline, of course, there are many other alternatives that takes place in nature. But if it's happening with a dramatic decline, like in vulture case, nature needs some time. Nature needs some time to cope up with the consequences and take some place and find out the alternatives on long scale of period of time. But when we talk about this cascade, as a one health practitioner, as a veterinarian rather, I would say that there are many challenges when we start implementation of this one health strategy on ground, and how to overcome it. I mean, that let us see what can be done, what we need to be, where to start with. So, in some of the cases, even in the landscape where we are working on, where we are starting the One Health Center, which has some of the facilities, and we are trying to get it functional with the help of concerned forest department. So, how it should be as a veterinary infrastructure facility, what it should include. So it should include treatment facility, operation theaters, animal keeping facility, quarantine areas. If the animal dies, we need to conduct the post-mortem in very, very scientific and very systematic and serious way. Then the sample collection kit should be available on ground in the center. Then preservative facilitation, then the transportation facilities, the team, the entire team, skilled veterinarians, skilled biologists should be available with this center, and they should have a basic idea about the wildlife health surveillance system, how it should do, how the active surveillance can be done, how the passive surveillance should be done, how the samples are collected, how they are preserved, how they are going to be transported. The data being generated from this all information should be interpreted and it should be analyzed and it should be presented at different level, increasing the chances of collaboration with different institutions, biologists, zoologists, veterinarians, policymakers as well. So basically, when we talk about this implementation, I'm talking about the One Health Center with focus on veterinary facility, but we are also trying to collaborate with some of the institutions where they are trying to collect the samples from the environment. So they are also trying to collect the samples of soil, water, vegetation, and so many other things. So basically, that One Health Center on ground will be very effective in different ways. It will be acting as a disease surveillance model. It will be acting as treatment facility, sample collection facility for free-ranging animals, and it would be included to dissipate the information to the right candidate, to the right stakeholder, and make the policymakers believe that One Health strategy is very, very, very important for near future. So what should be the way ahead? I mean, how it should be implemented. So some of the experiences where we started detecting the diseases, for example, when we were working in a couple of tiger reserves where we were collecting the samples for canine distemper. So the disease was detected in some of the leopards, and the department and the authority was very concerned that whether it's spreading a lot because some domestic counterparts. So we started detecting the disease with collecting the samples from domestic dogs, and now we have collected more than 1,200 samples, out of which almost more than 20 to 25% samples are positive for canine distemper. So when we talk about the host, there are many hosts which are maintenance hosts which keep the disease spreading. There are spillover hosts which spreads the disease and reproduce the disease to at least one cases with external factors. And there are dead end hosts which do not transmit the disease, but they will carry it for long period till death maybe. So this host has to be identified, and prevention strategies should be focused on that host. So in case of this canine distemper, where we were collecting the samples from some of the villages around the tiger reserves, so we focused that this disease should be detected first. Now the second step was controlling and prevention of that particular disease, so canine distemper. So we started immunization, mass vaccination drive in that area. We are still doing it in Tadobandhari National Park, Tadobandhari Tiger Reserve from Chandrapur district, Maharashtra. So where we are focused on vaccination of almost 3,000 dogs for rabies and canine distemper. But beside that, we started implementation of this wildlife disease risk analysis modeling. So WDRA includes the analysis, collecting the data, analysis of the data which is being observed by the authorities, and predicting the future outbreaks and the future scenario with number of mortalities, morbidity and mortality. So I understand there are a number of factors which affects the disease at any given time, which includes natality, means birth rate, mortality, means death rate, then immigration and immigration, I mean to and fro movement of the animals outside and inside this landscape. So wildlife disease risk analysis allows you to predict the extent of the disease, extent of the mortalities, and allow us to focus on prioritized landscape or the prioritized species in particular season, in particular duration at the particular area. In short, we get a better understanding of spatial and temporal distribution of the disease in terms of prevention strategies. So I understand that vaccination or immunization does not entirely stop the infection, but I'm sure that it definitely reduces the infectiousness of the infectious agent, and it definitely causes some reduction in the disease transmission and the disease spreading up to certain extent. So this is going to be a big strategy. So again, I mean, when we were talking about early warning system like WDRA, we need to be better prepared for vaccination as well. Then as a implementation authority or as a ground worker, I strongly feel that we need to have collaboration with different organizations. At global level, there are World Health Organization, World Organization for Animal Health, ministries, which includes forest ministries and even animal husbandry department, NGOs, and other stakeholders, mainly villagers and the communities living around the protected areas. So what we have started doing in some of the tiger reserves in central India, that we are working with several institutions, include Wildlife Institute of India, the multinational companies, the grantees, CSR funding agencies, the local NGOs and the communities, so that we can implement some of the parameters which are being focused on One Health practice and One Health approach. And now when the conservation scenario is teaching us a lot, that we need to involve the local community, the local stakeholder with us, otherwise any conservation measure is going to be failure. So what we were talking about, the awareness and the capacity building of the frontline staff, that includes the forest department and the animal husbandry department and the local NGOs, protecting the habitat, the biosecurity measures that has to be taken, and as discussed earlier, the vaccination, which helps us to control the spread of the disease up to certain extent, to reduce the number of the maintenance host in disease control way, and in some of the cases where the disease are found in just like the outbreak, so vaccination may not be the right strategy, but with WDRA modeling we can definitely predict the outbreak and its extent. So yes, this is what we have been doing, and this is the way ahead. I look forward that as one health practitioner for disease control, we are starting the same recommendations that are being suggested by WHO and WOH regarding awareness in schools and colleges and the villages, and the capacity building of the frontline staff, forest department, animal husbandry department, NGOs, and the community is happening on ground, and we have reached more than 5,000 people in this year so far. Vaccination, we have almost covered almost 2,000 animals so far. In the past, when we were trying to deal FMD outbreak in Gaur, we had also supported the forest department and animal husbandry department to cover the vaccination of cattle, of 10,000 cattle in the buffer villages of the tiger reserves. So it's a big number that helps us to control the disease and control the outbreak as well. And we are planning to start one health center at different locations where we what we discussed earlier, we are trying to implement it on ground. I absolutely understand it's very challenging, it's very daunting task, but what we have learned so far that if we take a step at a time, we can definitely implement one health strategy or one health approach on ground as a veterinarian. So that's it from my side. You may like to contact if you have any question further. You definitely please stay in touch. We would be more happy to collaborate and associate with different institutions to make it happen. And I thank you everyone for your kind attention. I'd like to have questions if any, please.
Thank you, Dr. Baba Bhaskar, for the wonderful presentation. And I think there are some comments and also some questions for you. Maybe I'll just read it for recording purpose so that it will be.
Please.
There's a trade-off competition between vultures and dogs. As we see increases in dogs at apotheosis, vultures decline. There are two ways to look at that. Vultures are efficient at eating carrion and regulate dogs. However, once dogs are established, they outcome vultures. This is a comment from Dr. Bipin Prajapati.
Yes, absolutely. That happen in many landscape that because of the dog population, vultures are not getting closer to the carcass, and we have also reported the same thing, that there are many dogs that they are trying to shoo away the vultures, and that's why the vultures are not getting the food. Absolutely, I agree. It's both way. It's both way.
A question from Dr. Asad. As you're working in the field of conservation, what do you think is good if there is breeding in zoo or private wildlife farm breed zoo animals? Will that come in conservation or it's illegal? That's the question. Yeah, anyway, you can.
Okay, I understand. So the basic concern is about ethical consideration. Then certainly keeping the animals in zoo would not be ethical if they are releasable. So most of the time the breeding centers or the conservation breeding centers are established with an objective that the animals which are captive bred are going to be released back to the wild or back to their natural habitat when the number increases. So conservation breeding centers are something not, it's not just a fantasy or anything like that. It's the need of some of the species. For example, vulture. Vulture were declining because of the toxicity of non-steroidal anti-inflammatory drugs. Had it not been taken into consideration to, you know, complete ban of these NSAIDs or painkillers, we cannot release these captive bred animals and the vultures particularly into its natural habitat unless the threat which caused the decline gets vanished. So we cannot, you know, we are catching the animals, we are keeping those vultures in the breeding centers. It sounds unethical or emotionally it's disturbing. But if we do not so, if we do not do that way, anyway the vulture as a species is going to extinct. But if we try to breed them in captivity, eliminate the threat from the habitat or the landscape which caused the decline in that vulture population, for example, painkillers. So now if the painkillers are not available, they are not visibly used, and there are reports that, okay, the ban is effectively implemented, then we can release those breeding centered animals or the captive bred animals or vultures to the habitat. So now they are going to increase their number in the wild. So in that way, I look at it, conservation breeding centers as a, you know, way forward to decline the status of some of the critically endangered animals to endangered species. So we need to improvise our conservation breeding center, which is way expensive and it needs skilled manpower as well. But wherever there is a threatened species, it can definitely help us to bring that animal from extinction to normal status for sure. And I am strongly with the conservation breeding centers in that way.
Thank you, Dr. Baba, with great appreciation from Nirupama for your presentation. There's a question. Have you seen IBR in Gore?
IBR?
Yeah.
No, so far.
Yeah.
There were some of the cases of bovine spongiform encephalitis, but they were not being reported because it's a notifiable disease. So, yeah, I mean, there are certain incidences where we also reported avian influenza in crow, where we found more than 1,000 birds died, but we could not report it because being a notifiable disease, that would have closed the poultry industry around 10-kilometer radius area of that location, which was not economically feasible. So the authorities asked the department that they should not release the report of positive post-mortem findings of the bird flu. But I mean, there are many incidences where we could not report some of the diseases, even though they are being found.
Yeah. Thank you. There's one more question from Savani Bhaviskar. How can we protect urban animals from diseases as common man? Question.
How we can protect?
How we can protect urban animals from diseases from common man point of view.
Okay. So vaccination is the best strategy when we are preventing the disease transmission. As we discussed, that reduction of transmission in the maintenance host, that should be the strategy. So yes, vaccination is the best strategy.
Yeah. Thank you. There is one more question, from Dr. Bipin. Are there incidents of FMD reported in Thadia areas in Gors?
Which area?
Tadiya.
Tadia. I don't know Tadia area, but yes, we have reported FMD in Gors from Navega and Nagzira.
Sorry, Tadoba. Wrongly spelled. Now it's
Okay. No. From Tadoba there were no incidences, but from the surrounding landscape where we were working in 2000 and 2003, there were 8 gores which were affected by FMD. Yes, there are reports of FMD in gores.
Yeah.
Not from Tadoba though.
Yeah. Okay. Thank you, Dr. Bahar Bhavishkar, for the wonderful presentation. And then if there is any more question, I think there is one question, maybe you can answer this also. This is going to be the last one. Is there any protocol to eradicate? I think it's written as eradicate CDV.
Okay. So, eradication, there are three strategies of disease dealing. So, one is control, second is prevention, and third is eradication. So, eradication of any disease has to be very specific long-term strategy for any department, which is almost, I would say, humongous task for any authority. So, I would say rather it should be a preventive strategy, and it would be always experimental designing. That it won't be possible for any authority to control and prevent that disease 100%. But yes, we can reduce the chances of spreading the disease by more than 85 to 90%. So I would not say that there is any protocol for eradication of CDV, but yes, there are protocols, and vaccination is one of the most effective protocol for prevention of canine distemper, which is aerosol transmission disease.
Okay.
I could see there is a report of FMD spray to elephants. From my landscape, for sure, we did not have any report of FMD in elephants.
The Sri Lanka, yeah. It's a comment saying that, clinically reported. This is with regard to FMD, I suppose. Clinically reported one case from FMD in a captive elephant subadult, one in Gorumara National Park. That's information given by Dr. Swati Mandal. Okay. And there is one question from Dr. Azad. Vets working with big cats, reserves and ungulates. Oh, he's asking whether anybody is just working, or if you have any information to just give about the people who are working on big cats, reserves, and ungulates in your region. Maybe you can just give some points.
Yes, we do work on big cats as well. That work is in association with the forest department. So yes, we do on tiger and leopards, and we do perform the rescue operations. We do also get involved in conflict mitigation, and we are talking to communities. And when there is a accidental case of mortality of these animals, we are reaching there as representative of National Tiger Conservation, the postmortem, and collect the samples as per the standardized operating procedure for big cats. So yes, and there are many ways. Dr. Khobragade, who is working in Tadoba, Andhari Tiger Reserve, and there are many ways actually. So if you would like to have contact, you can definitely contact us. We can provide all the names of veterinarians who are working in this landscape in central India on tigers and big cats particularly. Yes.
I think that's all for the time being. If there is any questions, maybe we'll just email you, and then it will be just circulated among the participants. Okay?
Thank you.
Thank you, Dr. Baha, for a wonderful presentation. Now I just call Heidi to just introduce the next speaker. Thank you.
Thank you very much. Excuse me. Thank you very much, Dr. Bad. That was very interesting. And now I will introduce the last speaker of this webinar today. It's Dr. Wahdi Azmi from Indonesia. Dr. Wahdi is a veterinarian working for wildlife conservation, especially elephants. He has over 26 years of experience in biodiversity conservation, sustainable development, and environmental management. Dr. Wahdi has received the Whitley Award as one of the top 14 global conservationists in 2021. The Whitley Award, known as the Green Oscar, is an international prize awarded annually by the Whitley Fund for Nature that recognizes outstanding conservation leaders worldwide. Dr. Wahdi has also been instrumental in developing elephant conservation projects in Sumatra, such as the Conservation Response Units. Today's presentation from Dr. Wahdi will be on when the umbrella species falls ill, disease risks for wildlife sharing elephant habitats. Dr. Wahdi?
Thank you, Heidi. Sorry. Yeah. Let me share the screen. Okay, thank you. Good morning, good afternoon, good evening, maybe good night for Heidi. Everyone, my name is Wahdi Azmi, and I'm honored to be here today to discuss an important aspect of wildlife conservation, disease transmission risk affecting Sumatran elephant. I would like to thank the organizer of this event, as well as Asian Elephant Support and Life Trust of India, and all partners involved. Today I will talk around how elephant as an umbrella species influence ecosystem health and how the interaction with humans, livestock, and captive populations impact the spread of the disease. Before I talk further, I'd just like to give some background to all of you that the distribution of elephant in Sumatra. So in fact, the red area, that shape area, is the elephant population's distribution, and the green one is the remaining forest cover, and actually the yellow area is the plantation area. So you can see in the middle of Sumatra and go to the south, most of the elephant population is actually places not really inside the green one, but in the middle of the yellow one. Means that the elephants is have more interaction with the plantation area. This is a map of Aceh province. The red dots indicate the distribution of human-elephant conflict, and as you can see, they are spread widely across the region. This clearly shows that much of Aceh terrestrial landscape is home to the elephants, and as the consequences, human-elephant interactions are widespread. The extensive overlap between elephant habitat and human activities highlight the urgency of addressing human-elephant conflict as both conservation and community challenge. Next slide. Aceh province is also home to the most significant elephant population in Sumatra. It still retains 3.5 million hectares of continuous forest. But it's not just elephant, but Aceh is the last stronghold for Sumatra typical endangered species, including Sumatran tiger, Sumatran orang-utan, and Sumatran rhino. With all these species facing extreme threat to the survival, Aceh remains one of the most crucial landscape for conservation effort, where protecting elephant means safeguarding an entire ecosystem. Next slide. Human-elephant conflict is often seen as just a livelihood issue centered on crop destruction and economic loss. However, this perspective is dangerously incomplete. When elephants enter farmland, they do not just damage crops, they come into contact with livestock and humans, exposing themselves to diseases, and they may carry deep into the forest. This is not only a threat to the affected elephants, but also serious ecological crisis. Many other elephants in the wild, along with countless wildlife species that share the same habitat, are at risk of infections. If we fail to address human-elephant conflict seriously, we are not just failing the community, but also we are endangering entire ecosystem. Conservation group must recognize that mitigating human-elephant conflict is not just about preventing economic loss. It is about protecting the integrity of Sumatran last remaining wild space and ensuring the survival of key species. Next slide, please. Sumatran elephant have large home range, and GPS tracking reveals that they travel between forest and agriculture area daily. This movement means they frequently come into contact with livestock, increasing the risk of pathogen transmissions. The longer the home range overlaps with human settlements, the higher, of course, the exposure risk. Next slide. A study conducted in Aceh found that elephant home ranges expanded significantly. They also expanded between 2019 and 2021, as elephant increasingly move between shrubland and farmland. Their contact with livestock and human waste also rises. This growing overlap not only intensifies human-elephant conflict but also heightens the risk of disease transmissions, posing a serious threat to both wildlife and human community. Next. The GPS collaring provides essential data for managing human-elephant conflict more efficiently. It confirms elephant home range and movement pattern, helping us understanding where and how they travel, which is the key to identifying high-risk conflict zone. It supports well-informed barrier strategies by guiding the placement of effective large-scale barrier that prevent crop damage while maintaining habitat connectivity. It also acts as an early warning system. Next slide, please. Can you push the can you play the animation? Yeah, this is a sample of animation of GPS collect data illustrating elephant movement pattern. As you can see, the majority of their movement occurs in community land rather than in the primary forest. This highlights the significant overlap between elephant home ranges and human settlements, reinforcing the urgent need for effective conflict mitigation strategy. Next slide. So this graph from GPS collar data just an illustration that both elephant and human tend to prefer the same type of landscape, of course the flat one and the lowland area. Unfortunately, when spatial planning takes place, human needs are almost always prioritized, leaving elephant with increasingly limited habitat. This overlap intensifies human-elephant conflict and highlight the need for more balanced land use planning. Next, please. Several factors contribute to the spread of the disease in elephant habitat. Free-ranging livestock often graze near the elephant corridors, increasing the likelihood of transmissions. In addition, captive elephants that interact with wild population can also spread disease such as EHP, contaminated water, so another major concern. And finally, human-elephant interaction in tourism area and conservation activities create additional opportunities for disease transmissions. Next, please. As you can see in this table, elephants are susceptible to various bacterial, viral, parasitic disease. Some of these, like tuberculosis and brucellosis, are transmitted from livestock. Others, like EHV, pose a significant threat to young elephant in both wild and also captive elephant. If an infected elephant carries this pathogen into the forest, they may affect other species, including various wildlife. Next, please. To reduce disease risk, we must rethink how we manage livestock and elephant interactions. There's many community in the edge of forest, where the elephant also live coexistently, but the community keep the livestock like free roaming. So it's suggested to reduce the risk. We must enclosing the livestock, implementing better landscape scale barrier, and strengthening disease surveillance are all key solutions. Additionally, improving biosecurity measures in captive elephant facility will help control the spread of the disease. Continue. A more effective strategy for mitigating human-elephant conflict is implementing landscape-scale barrier instead of only fence individual crop plots or farms. So we can use natural barrier, of course, as following the natural barrier formation to be more efficient and at the same time also effective as a large scale solutions. Continue. Can you play the animations? So this is the, oh, sorry, just continue. Just, this is just to inform the different elephant corridor. Next one. Next. Yeah, this is the example of the elephant movement from GPS data, showing that elephant also restricted the movement by the extreme existing natural barrier, but also at the same time the human spread and development also actually impacted or restricted by the extreme topography. So this in this map, the dots, the blue one is actually the data of elephant distributions, and the yellow one is showing the extreme slope, and it shows that there are some existing natural barrier that keep the elephants in one area and separate to the community. Continue. This is the example, why we need to consider the existing natural barrier to be more efficient in the way to for our barrier strategy, and also at the same time a bit more effective because we don't have to barrier the entire area, but also in gaps where the elephants are passing the existing natural barrier. Continue. This is the example of the barrier strategy in Aceh, where we not only barrier each of the crop plots, but rather in the landscape level. So it's more efficient, the investment, because it can protect larger community instead of only the one who has the capital to build the barrier themselves. Yeah. So it's like example of the artificial barrier in the area where the gaps of existing natural barrier occurs, and we put the artificial barrier. Continue. Yeah, the power fence. Continue. Yeah, this is also an example of combination of the canal and also the power fence in one of the gap of the existing natural barrier in Benue area. And it's help to reduce the interaction between the wild elephant and also the community area, where they also keep the wild the livestock with them. Continue. Next. This is just an example of after we put the barrier in some areas, when we study, we have some reduction of the human conflict incident of the barrier strategy applied at landscape level. Can you click one time? Yeah, this is to show the reduction of human conflict in Aceh. Next. So in conclusion, the conservation of elephant must go beyond reducing human elephant conflict. We need integrate strategies that address disease transmission risk, strengthen land use planning, and also prioritize biosecurity. I think that's all the slide, Heidi. I will thank you very much for the attention.
Thank you, Wahidi, for the presentation. Any questions? If you have participants, if you have any questions for Wahidi, you are most welcome to just type in the chat box. If not, I request Heidi to just continue with the rest of the proceedings. Thank you.
Thank you. Well, if there are no questions, that comes to the end of this session and this webinar. Thank you very much, Dr. Wahidi, for your presentation. And we hope that you all will join in the next one, which will be 2 months from now. So again, if you have any follow-up questions, please feel free to contact us, either Daniel or I, and we will pass them on to the presenters. So thank you very much, everyone, and hope to see you next time.
Thank you, Daniel. Thank you, Heidi.
Thank you.